Okay, welcome back students to Ram Sarki class. Today we are going to study about the internal resistance of a chemical cell or simply a cell. So what it is, how it affects the flow of current and on what factors it depends. So today in this video we shall discuss about all those main things. So first let's understand what is internal resistance. This is mainly a part of chemistry, so we are not going very deep into the idea of cell and its different properties, because the main theme of this lecture is to discuss about the internal resistance of the cell. So as you know, this cell is formed by placing electrolyte in a voltmeter, and then we emerge two metallic rods depending on the nature and what sort of cells we have to make. And one terminal becomes positive, another becomes negative with respect to the solution or electrolyte inside the voltmeter. This device is known as voltmeter. Please mind that voltmeter and voltmeter both are different things. So a voltmeter is what? It measures what? Potential. It measures potential, difference between 2 points in an electrical circuit. But a voltmeter, mind it, voltmeter. Voltmeter is a device on which, or you can say it's a container in which electrolyte is placed and in which two chemical or electrodes are formed. One plays as positive and another becomes negative. So this is simply a structure of chemical cell. Now what happens when you connect this through a external resistance? You must be seeing this is external resistance. These are the two ends of connector. It is symbolically denoted as this. We all are very aware of this. Okay. So if this is connected to a resistance, as I done earlier, so this is my external resistance and this is the same resistance R. So when this happens, the current flows from the positive terminal to the negative terminal, but outside the cell. We are not talking about the part inside the cell. Electric current flows from the positive end to the outer end in the external circuit. Okay, and that is the direction of conventional current. But what happens inside? The positive ions start gathering or accumulate just close to this terminal and making its potential more and more positive with respect to the solution. Simultaneously, that means the positive ions from the solution has gone to this particular rod and making it a positive, and this accumulation of positive charge particle producing a positive potential at this end. Simultaneously, the negative ions go to the other end or other rod and making it negative. So this becomes a negative rod, that is forming a negative potential on this right side rod and putting positive potential on this left hand side rod. So it means inside the cell, what happening? The positive terminals, the positive ions inside the cell goes from solution to the positive end and the vice versa. This is very simple and clear, and this is the circuit diagram for the so formed cell. So the current flows this way. Okay. Now what internal resistance is, let's understand what the definition speaks. The resistance provided by a cell due to the flow of current between the electrodes inside the cell is called its internal resistance. Okay, because the ions are moving from solution to the positive end, so they are creating or producing a current inside the cell. So this flow of current is offered by something that is obviously what the factors going to affect its properties or flow of current, and that resistance or that hindrance provided by the cell to the flow of the ions inside the chemical cell. Okay, so generally inside the electrolyte of a cell, the positive ion flow from the lower to the higher potential against any particular background, or you can say against the background of other ions. Okay, so this is how the current is opposed when the cell is formed, but this flow of current is opposed by something, and that something is known as its resistance or the internal resistance of the cell. Okay, that is called internal resistance of the cell. So the internal resistance of any chemical cell, as I told, denoted by R. So R simply depends on the following factors. They are very simple. First is nature of the electrolyte. Either it is ZnSO4 or copper sulfate, zinc sulfate, whatever, H2SO4. So these are the different types of solutions or electrolytes can be used. So it depends on the nature of the electrolyte. Next point is the internal resistance depends directly on the concentration of the electrolyte. If the concentration of the electrolyte is more, then R becomes larger. More concentration, R is becoming more and more. Okay. Next is, it is directly proportional to the distance between the two electrodes, that is the distance between the two rods. Say this is denoted by small d. If this distance is more, then ions have to travel more and more distance, and during that motion they will be experiencing more number of collisions with other ions, that is causing a kind of hindrance or resistance. So that internal resistance will increase more the distance between the two rods. Very simple. Next is, the internal resistance is inversely proportional to the area of the plates well immersed inside the liquid. More part of the electrode immersed in the liquid, liquid means electrolyte, then R will be less. So A is large, then R will be less. These are common symbols. Please don't get confused. Area is what? The area of the plates or portion of the plates that is immersed inside the electrolyte. At last, when the temperature rises, then the solution becomes little diluted. And in this situation, the ions can easily go from one to other part of the electrolyte, that is decreasing the internal resistance. So when you increase the temperature of a cell, its internal resistance simply decreases. So dear students, this is the simple statement of internal resistance and its few properties. In examination, generally the MCQs are asked what happens to the if concentration is increased and if the distance is increased. So like one question, the internal resistance of a cell will increase when? Our internal resistance of the cell will not increase when concentration is increased, distance between the electrode is reduced, area of the plate is reduced, and temperature is increased. So out of these four parameters, most of the time we get confused in the examination. So be well aware of these four factors, because nature plays its own part, but for any particular chemical solution, that is for any particular electrolyte, the last 4 points are very important. If we ignore the type of electrolyte, we are not going to discuss what either it is H2SO4, CuSO4, ZnSO4, whatever. So, but the last four points are very important. Okay, so this is what internal resistance of any chemical cell. Okay, so this is the resistance provided by the nature or the electrolyte inside the cell to the flow of its current is known as internal resistance, denoted by a small r. So now we shall discuss what is the relation between the terminal potential of a cell and the EMF of the cell and internal resistance. These three symbols will be put together in a single formula, and that will form a new basis. Okay.